students, today we will start with a new topic, that is biomolecules. What are biomolecules? The molecules that frame out a living organism's body. We'll write one sentence here regarding biomolecules. The molecules that are constituents of living organism's body. In general, we know that molecules are made up of cells. So, and whatever the biomolecules that are present in one living cell are termed as cellular pool. All biomolecules in a living cell or one cell, also we can use a term, living cell, are termed as cellular pool. Cellular pool. It means all biomolecules in a living cell are termed as cellular pool. Biomolecules are of two types. One is organic form and second one is inorganic form of biomolecules. Under organic form, we can take examples like proteins, carbohydrates, lipids, nucleic acids. Under inorganic form of biomolecules, we can take examples like minerals or elements and water. Can we extract these biomolecules? Means answer will be yes, by using trichloroacetic acid. But the percentage or concentration of trichloroacetic acid used to extract these biomolecules, percentage will be different. To extract biomolecules, trichloroacetic acid this compound is used, or this solution is used to extract biomolecules. How? The question is how means trichloroacetic acid is added with the tissue of living organism and these both are grinded. Then the obtained total mixture is termed as slurry. The grinded mixture. Now, this slurry is allowed for filtration, is filtered. Then, the obtained, whatever the solution is there, we will divide that one into two different parts or two different fractions. After filtration, we get two parts. For suppose assuming that this is a test tube or the beaker in which we are taking the filtered slurry, and we are using a net here which is having some holes like this. Okay. So whatever the slurry is there, that we are putting here. Then this is called as slurry. But after filtration through the net, some solution or liquid will be falling down out of the filtering net. Since the total tissue or living tissue of animal or a plant body is mixed with acid, some of the acid will be coming down to the bottom of the test tube. So after filtration, we call this one as filtrate. Filtrate. Then what is left over here? The leftover part is called as retentate. This retentate is also the term is according to NCERT only. Whatever the part that is left over is called as retentate. Whatever the solution that has come out of the net from the slurry is called as filtrate. Since the filtrate that is obtained after filtration through the net is because of soluble or mixing or dissolving in the trichloroacetic acid, this can also be called as acid soluble pool. Again I am repeating. We have got after filtration of the slurry two parts. One that is left over in the net, that we are giving a term as retentate. The one which got dropped out of the retentate because of the acidic nature or the acid that we are taking, trichloroacetic acid, and some of the particles from the slurry are mixed with or dissolved in the acid and got filtered to the bottom or out of the filtering net. So we are calling the filtrate as also acid soluble pool. Now coming to the molecular weights. Molecular weights of biomolecules in retentate. Approximately are 10,000 Daltons. Means whatever the biomolecules that are present in the retentate, their molecular weight will be approximately of 10,000 Daltons of weight. So Daltons is the units. Now when we are seeing the molecular weight present in the retentate, let's consider the molecular weight of the molecules whatever are got dissolved in the trichloroacetic acid and came into acid soluble pool. Molecular weight of particles or biomolecules in acid soluble pool. It was seen like only between 800, means approximately near to 800 Daltons of molecular weight, is approximately 800, between 800 to 1000 we can take Daltons of molecular weight. And if we take here examples under the particles that are having molecular weight of nearly 10,000 Daltons, they are considered to be proteins, carbohydrates, nucleic acids, very much negligible lipids. But here, if you take example, what type of biomolecules have been got down into the filter rate, we are seeing lipids majorly. But lipids also we are studying under the large macromolecules only, macromolecules. But how can we consider this one? How can we differentiate the lipids that have been entered into the filter rate and the part of lipids that are present in the retentate? This point we can learn as, according to size of particle, we'll take here biomolecules, size of biomolecules, the order will be carbohydrates are having larger size, greater than lipids, greater than nucleic acids, greater than proteins. This is the order. But there is another order that is different from this sequence, that is carbohydrates greater than lipids, this one. That is percentage of biomolecules in a cellular mass or a tissue. Cellular mass or some tissue. Again here the order changes. The first thing we have seen, basing on the size of the biomolecules, carbohydrates greater than, lipids greater than, nucleic acids greater than, proteins. But here the sequence changes, that is proteins greater than. First priority goes to one type of molecule, that is water molecule. Next priority goes to proteins, but since we are discussing about only biomolecules here, I'm writing only as proteins greater than carbohydrates, greater than lipids, greater than nucleic acids, greater than lipids, greater than nucleic acids. This second order is basing on the percentage or the quantity we can consider. So the next priority goes to ions that I am not mentioning here. So the order will be changed basing on the type of question. Again I am telling, basing on the size of the biomolecule, the first order will be like this: carbohydrates, next lipids, next nucleic acids, next proteins. But basing on how much percentage of biomolecule is present in the tissue that we have taken. For suppose if you have taken 100 grams of tissue, any living tissue, then again the order changes. In that tissue, the protein content will be more. Next priority will be to carbohydrates. Next will be to lipids. Next will be to nucleic acids. So in our NCERT, they have given what are the percentages in page number 147. Students, you can go through that once. But sometimes what happens, the examiner will not mention anything. Just they will ask ascending order or descending order. Then we have to opt for such type of answers. Till we have seen what are the types of macromolecules that are present in the retentate. Next, what about the lipids? Here, the molecular weight of the molecules that are present in the retentate are considered to be having 10,000 Daltons, and here it is considered to be only one type of lipid will be filtered through the acid, that is trichloroacetic acid. Now, lipids based on size are considered as macromolecules, because based on size, the second priority of the total biomolecules goes to lipids. But lipids based on molecular weight, I am mentioning like this, are considered to be micromolecules. So this is the important point regarding the lipids that are filtered into trichloroacetic acid or the filtrate or acid soluble pool. Next, coming to order of what type of biomolecules are synthesized by different types of living organisms in abundance. Bacteria are considered to be heavily synthesizing living organisms of biomolecules. Next priority goes to algae. Next priority goes to plants. First thing is, we can consider cyanobacteria type of organisms, because the smaller the size of the organism, larger will be the synthesis of the products. So when we go for the priority of abundant biomolecule synthesis among different living organisms, then first priority goes to bacteria, which synthesizes more content of biomolecules, like carbohydrates we can tell. So I will frame out that line also. Order of carbohydrate synthesizing organisms. So here we will write abundance. Order of carbohydrate synthesizing organisms based on abundance. So here answer will be, first priority goes to bacteria, second priority goes to algae, second priority goes to plants. Here I am giving you some of the important orders also that will be useful for your examination point. Carbohydrates are defined as hydrates of carbons. How can we analyze this definition? Carbohydrates in general are represented as H2O. This is considered as general formula of carbohydrates. Now, if we see the ratios, this is only 1 in number, carbon. Hydrogens are 2 in number. Oxygen atom is again only 1 in a number. Carbon is 1 in a number. Hydrogens are 2 in number. Oxygen atom is 1 in number. So we can give out the ratios of carbon is to hydrogen is to oxygen as 1 is to 2 is to 1. But if a question is asked, what are the percentages of those 3 important elements in human body, again the order changes. Order of abundance of elements like carbon, hydrogen, oxygen in human body. The percentages are given in NCERT. Basing on the percentages, oxygen content is more than carbohydrates. Again, carbohydrates contents are more comparing to the hydrogen. If we go for values, oxygen constitutes of 5.5 percentage, 65.5 percentage. Carbon constitutes of 18.5 percentage and hydrogen constitutes of 0.5 percentage in human body. So again here this will be the answer. But coming to the sequence of ratios in the general carbohydrate molecule, it will be 1 is to 2 is to 1. But the ratios or the order will be changed, percentages will be changed when we are talking about the order or the percentage of elements in the human body. Now, I gave you definition as carbohydrates are nothing but hydrates of carbon. How can we analyze that? One means this is water molecule, nothing but called also considered as a hydrated compound, and this is one. So if I write here like this, carbohydrates are considered as hydrates of carbon. This simple word I have written in the form of sentence. Carbohydrates are hydrates of carbon. Okay. Now, there are further classified points regarding these carbohydrates. We can write that one basing on the number of molecules that are termed as basing on the number of units. Okay, so based on number of units, carbohydrates are considered to be, first one, monosaccharides. They are made up of only one unit. What is meant by saccharide? Saccharide means sweet. Mono means one. So one sweet unit we can consider. Okay. So one unit. Here examples we can take as glucose or fructose or galactose. All these are three. This is generally present and responsible for our body, means energy giving molecule in our body. Fructose, the normal sugar that is present in the fruits. Galactose, we read this one is existed in the liver. And also one of the compound of lactose molecule. So when one unit is behaving as one complete molecule, then we are giving a term as monosaccharide. Then another one, disaccharide, means when two units constitute as one molecule. Disaccharide, it is made up of two units or having two units. Here example we can take as maltose, which is made up of two glucose units, sucrose, which is made up of one glucose, one fructose molecule, and we can also take lactose, which is made up of one glucose and one galactose molecules. Means it is made up of two units, again two units, again two units. Next. Third example we can take as oligosaccharides. It means that they are made up of more than 2 or more than 3 number of units. Up to 8 we can consider oligosaccharides made of more than 3 units of monosaccharides. So here we can take example as raffinose, Next, polysaccharides. We can take example as starch, cellulose, etc. Fourth one, polysaccharides, means many number of monosaccharides will be constituting as one complete functional molecule. Polysaccharides made of many monosaccharides. Here we can take examples like starch and cellulose. Now, so basing on number of carbons in these carbohydrates, again the terms changes. For suppose monosaccharide examples, here we have given as glucose. Glucose is made up of 6 carbons. Fructose is also made up of 6 carbons. Galactose also made up of 6 carbons. Likewise, basing on the number of carbons in a carbohydrate, because here we have given here one example of carbohydrate as CH2O. If carbon is 1, then the term differs. We can use a term like monomer. If carbon numbers are 2, then again the term changes. So, not monomer. If there is 1 carbon, just we'll say that it is a general carbohydrate name. Okay. So, basing on the number functional activity of the carbohydrate, it will start from addition of 3 carbons, means from trioses. So, there are some types of carbohydrates like acetyl coenzyme, etc. But mostly they are considered as one of the intermediates in the reaction. But according to our syllabus, we will read from trioses onwards. First one, if there is only 1 carbon, I said you will start with 3 carbons, right? Okay. 3 carbons, then according to the ratio of CH2O, there must be double the number of hydrogens. So it will be 6. Next, again, oxygen ratio has to be only 1. So again, it will be 3 only. If the formula is like this, then we are telling this one is having 3 carbons, or this compound is having 3 carbons. Then the term given is triose, which we read mostly in respiration chapter while reading the catabolic activities or the metabolisms. Triose. Here we can take examples like in glycolysis we see, and in C3 cycle also we see some of the examples like PGA, phosphoglyceric acid, or another example glyceraldehyde 3-phosphate. Another example dihydroxyacetone phosphate. Full forms of this are nothing but PGA means phosphoglyceric acid. GLY-3-P means, PGA means phosphoglyceric acid. This one, second one, GLY-3-P means glyceraldehyde 3-phosphate. Next, this is dihydroxyacetone phosphate. Actually, this phosphoglyceric acid is considered as acidic form. Likewise, you can remember acidic form because in the name itself we are seeing phosphoglyceric acid. Okay. Second one, glyceraldehyde 3-phosphate. Here it is aldehyde form. And DHEAP or dihydroxyacetone phosphate. This is nothing but ketone form. All these forms we will see. First one we will see in C3 cycle as the first formed 3 carbon compound. These both we will read in glycolysis. And this one we also see in Krebs cycle also in a respiration chapter. So, second example, when we take 4 number of carbons, then again hydrogen has to be double, oxygen has to be 4 in number. We give a term as tetrosis. So, I will write here 4 carbons. The name given is tetrosis. In C3 cycle, during the study of regeneration step, in C3 cycle we have 3 steps: carboxylation, reduction, regeneration. When we are reading about regeneration, then we will come across one 4 carbon compound, that is erythrose. Example, erythrose. Erythrose is a 4 carbon compound. Next, if carbon numbers are 5, then hydrogens has to be double, oxygen has to be again 5 in number. Then we give a term like this compound is having how many number of carbons? 5 carbons. Then we give a term like pentosis or pentose. So examples are ribose type of sugar or deoxyribose. Ribose is the pentose sugar in genetic material, that is RNA. In RNA we can see what is the type of pentose sugar means ribose type of pentose sugar means only 5 carbons are there. Coming to deoxyribose is the pentose sugar that is present in the DNA, genetic material. Next, fourth example. If the carbon numbers are 6, then hydrogens has to be 12 according to the 1:2:1 ratio of general carbohydrate formula. Again, oxygens also has to be 6. Means this compound is having how many carbons? 6 carbons. When the compound is having 6 carbons, what is the name that we are giving? Hexoses. Hexose. So what examples can we take? Already you are very much familiar, that is glucose having 6 carbon, fructose having 6 carbon, galactose having 6 carbons. Next, If the carbon number is 7, again hydrogens has to be double. Oxygen also has to be 7 only. It means that this compound is having how many carbons? 7 carbons. This is called as heptose. Then what example can we take here? Again, in the C3 cycle regeneration step, we have one intermediate compound there, that is pseudoheptulose 7-phosphate or pseudoheptulose 1,7 bi or diphosphate. We can write here example as pseudoheptulose. This is a 7-carbon compound which we will come across in the regeneration step of C3 cycle.